Sup nerds, it's Lindsay again. So been doing some more studying for the MCAT, and right now I'm working on some of the kind of basic biology like the cell and the cell structure and all that. So I was going to do the same sort of thing I did last time. I made flashcards, not nearly as many flashcards as I had in the last time I did this, but still I was going to just kind of go through the flashcards and then talk a little bit about them. So if that's the kind of thing you would like to do, then stick around. So the first one I have is cell theory. So it's the 4 tenets of cell theory. There are 4 tenets of it now. There were originally 3. The 3 original ones were all living things are composed of cells. The cell is the basic functional unit of life, and that cells arise only from pre-existing cells. The 4th one that's been added more recently as technology has improved and we've started to learn more is that cells carry genetic information in the form of DNA, and then they pass that from parent cell to daughter cell when they reproduce. So that's the 4th tenet that's been added. I don't think cell theory is necessarily considered high yield for the MCAT, but it was asked about on one of the Kaplan diagnostic tests. I do remember that. So I think it's just important to really just know those 4 tenets. I would say you probably don't need to know anything else about cell theory, like how those things, how it came to be that we learned those 4 things. I would say probably just know the 4 tenets. That's really all that matters. All right, so moving on into stuff that is a little more high yield, eukaryotic cells. So as we all know, if we took a biology 101 class, cells are grouped into prokaryotes and eukaryotes. For the most part, I would say it's more important that we know about eukaryotes for the MCAT, at least as far as memory serves me, but it is also important to know about prokaryotes as well, because that's things like viruses and bacteria. So that is stuff that is important for if we want to be doctors. We need to understand about those things. So starting with eukaryotes, really the difference between them, eukaryotic cells contain a blank while prokaryotic cells do not. That is a nucleus. So the eukaryotic cells are the ones that have the nucleus. There's other differences as well, but that's really the primary difference, is that the eukaryotic cells contain a nucleus and prokaryotic cells do not. They kind of just have that little bundle of DNA in them. They don't have a true nucleus. All right, next we have blank allow for the diffusion of molecules throughout the cell. So basically, what are all the organelles sitting around in? What allows for the diffusion? What are things like peroxisomes travel through as they're, or not peroxisomes, what was the thing I was thinking of? I can't think of the word right now. It's in one of the cards later, but what do things travel throughout the cell? They travel through the cytosol. That's kind of that sticky stuff that's all throughout the cell that the organelles are suspended in. I don't know if the rest of y'all ever had to make a diorama of a cell when you were younger. I did. I remember having to do that. DNA is organized into chromosomes. One of the things that's always kind of tripped me up a lot is a lot of the words when it comes to talking about cells that start with that prefix of like chrome. If you've got the chromosomes, you've got the chromatids, you've got all those things that sort of tripped me up. I always got confused with those, so I don't know if other people feel the same way, but DNA is organized into chromosomes. Blank in the nuclear membrane allows selective two-way exchange of material between cytoplasm and nucleus. Those are the nuclear pores. That's how things go into and out of the nucleus from the cytoplasm. Linear DNA is wound around organizing proteins called blank and further wound into linear strands called blank. So let's start with the first one. Linear DNA is wound around organizing proteins called histones and further wound into linear strands called chromosomes. Getting back to those chromosomes. Like I said before, that was something I kind of had trouble with, keeping straight the different chromosomes, chromatids, that sort of stuff. So there's quite a few flashcards about those. These flashcards really were stuff that I felt I needed to work on, and that's why I made flashcards of them. So if there's anything you feel like I skipped, it was probably just because I didn't make a flashcard for it, because I just didn't really feel like I needed to go over it again and again. That's the thing about studying for the MCAT. You really just need to be smart about the way that you study. If there's something that you know very well, you don't need to keep going over it over and over and over again. I mean, of course, you need to remind yourself occasionally because you don't want to forget it, but you don't need to be putting as much time into it as you are putting into things that are problem areas for you. There is a subsection of the nucleus called the nucleolus where blank is synthesized. So what goes on in the nucleolus? That is where we synthesize our RNA, or ribosomal RNA, is in the nucleolus. The mitochondrion contains 2 layers. This, thankfully, is one of those ones that is fairly descriptive. So the 2 layers of the mitochondrion are just the outer and inner membranes. No one strained any brain muscles on naming those, thankfully. The blank serves as a barrier between the cytosol and the inner environment of the mitochondrion. So that would be the outer membrane of the mitochondria. The blank contains the molecules and enzymes of the electron transport chain. You probably could guess based on what the last few questions were, but that is the inner membrane of the mitochondria. It contains the molecules and enzymes of the electron transport chain. I usually just abbreviate it ETC just to save myself some time. That's something that really comes up to haunt you a lot in biochemistry, is that electron transport chain. So it's good to get a good kind of basic foundation of understanding of it at the basic biology level, because you will definitely need to know a lot more about it once you get into biochemistry. The space between the inner and outer membrane is called the, once again, very kind of self-explanatory name, that is the inner membrane space. The space inside the inner membrane is called the, that's where we find the mitochondrial matrix. The pumping of protons from the mitochondrial matrix to the inner membrane space establishes the, it's another thing that'll kind of come back to haunt you in biochemistry, so it's good to understand. That's the proton motive force right there. I'm going through them a little faster because the last video I made like this ended up being an hour long, and so I'm sorry if this is a little too fast. I just don't want to make another hour-long video. Blank are membrane-bound structures containing hydrolytic enzymes that are capable of breaking down many structures. Now, I'm a big believer in understanding word roots in the sciences, because I think it really does help you, especially once you get to anatomy. It is so helpful to just know the word origins, because even if you come across a word that's very complicated, you can kind of break it down and figure out what it means. And I think this is one of the ones that you can definitely do that for lysosomes. If you're familiar with the word roots, that lys, that kind of means to break down or to cut, and then some just means like body, basically. So it is a little body, it's a little thingy that breaks things down. That's what it does. It sucks things up and breaks them down. Lysosomes often function in conjunction with blank, which transport, package, and sort material traveling to and from the membrane. I can't talk tonight. That is endosomes, those guys. Blank is utilized primarily for lipid synthesis and the detoxification of carbon drugs and I was looking at orgo earlier, so I've got carbon on my brain. Let me say this one again. Blank is utilized primarily for lipid synthesis and detoxification of certain drugs or poisons. That is the smooth ER, or a smooth endoplasmic reticulum. That's another one I usually abbreviate to save some time. The smooth endoplasmic reticulum transports proteins from the rough endoplasmic reticulum to the Golgi apparatus, another little structure within the cell. Blank function in the breakdown of long-chain fatty acids via beta oxidation, synthesis of phospholipids, and contain some of the enzymes involved in the pentose phosphate pathway. Another guy you're going to have to know in biochemistry. Those are the peroxisomes, and those guys are bodies. Remember, some, that means body, bodies that contain hydrogen peroxide. So that's how they get their name. Microfilaments are made up of solid polymerized rods of, you might remember learning about this in anatomy as well. It's actin. You talk a lot about actin and myosin when you get to anatomy because they're involved in muscle contractions. Actin filaments can use ATP to generate force for movement by interacting with, I just said it, with myosin. And the way that actually works is the myosin pulls on the actin. And the way I remember that this is involved a lot with muscles is because that prefix, MYO, that means muscle. So it pulls on the actin to cause muscle contractions. Microfilaments form a cleavage furrow in cytokinesis. That's one of the jobs that they do. Blank are hollow polymers of tubulin proteins. Those are microtubules. Blank are projections from a cell that are primarily involved in the movement of materials along the surface of the cell. Those are cilia. You see those a lot. One of the things that, I don't know if you're like me, but like me, I typically think about cilia as being part of like single-celled organisms, but another place where you find cilia is actually in the lining of your lungs. It's these little, they have these little cilia, your lung cells, that kind of move mucus and stuff along and kind of keep the mucus from settling on your lungs. So I think that's probably a pretty good thing to have, I would imagine. Yeah, kind of the entire respiratory tract has that cilia lining them, so that's a good thing. So the answer to that one is cilia. Cilia and flagella are composed of blank pairs of microtubules forming the outer ring and blank microtubules in the center. So this, if you remember, is that 9+2 rule. If you remember that from class, if I can get the card. The 9 and 2. They call it the 9+2 rule. And I was actually just flipping through this book because I knew that card was coming up. See if I can find the picture of it. Here it is. Nice picture. This picture of that 9+2 structure. All right, next card. Blank are for organizing centers for microtubules and are structured as 9 triplets of microtubules with a hollow center. Those are the centrioles. Centrioles are found in a region of the cell called the centrosomes. This is another one that can get a little tricky because there's a lot of words that sound kind of similar. The centrioles, and then they're found in the centrosome. So for me, the kind of stuff that helps with these, when words are just very similar, what helps is just redundancy of just going over again and again what the specific definition of each one is and what they do. That's how, that's the way I can distinguish them. I have trouble with, I actually generally enjoy mnemonic devices, but I have trouble with them when it comes to words that are very similar like that. When it comes to words like that, I typically end up just going for kind of brute memorization for learning them. During mitosis, the centrioles migrate to opposite poles of the dividing cell and organize the... So what do we get when those centrioles go to opposite ends of the pole? Do y'all remember this from mitosis? That's how they get the mitotic spindle, that little guy. The microtubules emanating from the centrioles attach to the chromosome via blank and exert force on the sister chromatids, pulling them apart. Those are the kinetochores. All right. Keratin, desmin, vimentin, and lamins are all Of those, the ones you're probably most familiar with is keratin. Those are all intermediate filaments. Blank cover the body and line its cavities, providing a means for protection against organs. So what's your skin made out of? What kind of tissue is that? That is an epithelial tissue. Epithelial cells are tightly joined to each other and to an underlying layer of connective tissue called the... It's another pretty kind of self-explanatory name. That's the basement membrane, and that's easy to remember because basement, of course, very bottom of a house, and that's the very bottom of the epithelial cells. In most organs, epithelial cells constitute the blank, or the functional parts of the organs. The parenchyma. That's a fun word to say, isn't it? Simple epithelia have blank layer of cells. I kind of gave it away with the singular there, but yeah, one layer of cells. Simple epithelia, one layer of cells. Then stratified epithelia have just multiple layers of cells. It can have several different numbers, but if it's simple, it's just one layer. If it's stratified, it's multiple. And then this kind of epithelia appear to have multiple layers because of difference in heights, but they actually just have one layer. What's the name for that? Well, that is called pseudostratified epithelium. So again, word origin, pseudo, so fake, pseudostratified epithelia. It looks like it's stratified, but it's actually simple. Blank supports the body and provides a framework for the epithelial cells to carry out their functions. What kind of tissue is that? That is connective tissue. Connective tissues are the main contributors to the stroma, or the support structure. So remember that connective tissues have a lot to do with support. Most cells in connective tissues produce and secrete materials such as collagen and elastin to form that's the extracellular matrix. Blank are single-celled organisms that are visually similar to bacteria, but contain genes and several metabolic pathways that are more similar to eukaryotes. So the three main domains of life, we've got the eukaryotes, and then we've got the bacteria, and then the other one is the archaea. Archaea, in my opinion, are very, very interesting because we originally thought they were just kind of a division of bacteria. We called them just like archaic bacteria, and then we are still really finding out that they have a lot more going on than that. They're actually much more similar to eukarya than we had originally thought. They're very, very interesting things. I find them to be really fascinating just because of the way they're what they call extremophiles, and that they can typically survive in very kind of extreme environments where we previously had thought no other life could survive, like those sort of boiling hot vents at the bottom of the ocean. That's where you see a lot of archaea, where or they're in environments with just kind of crazy temperatures and no light, and really under conditions that for the longest time we thought that life absolutely could not be sustained. And then the archaea were just like, hey, what's up? Archaea are notable for their ability to use alternative sources of energy, which is kind of like what I was talking about, that they can use chemical sources of energy rather than like solar sources of energy, like a lot of other organisms, or like source of energy from eating other organisms like us. All right, all bacteria contain a blank and blank. Some have blank or blank. So really just what do all bacteria have? So all bacteria have a cell membrane and cytoplasm, and then some may have flagella or fimbriae. Bacteria that require oxygen for metabolism are obligate aerobes. Remember, aerobic exercise, if you ever heard people talk about that, that's exercise that makes you breathe faster, that makes you suck in a lot of oxygen. So bacteria that require oxygen, they are obligate aerobes. Bacteria that use fermentation or some other form of cellular metabolism without oxygen are, those are anaerobes. So an, without oxygen. And then further within that division of anaerobes, we have a couple different categories. So anaerobes that cannot survive in an environment with oxygen. So anaerobes that absolutely cannot stand being around oxygen are called obligate anaerobes. And then bacteria that can switch between aerobic and anaerobic processes are called facultative anaerobes. So they're anaerobic, but they can switch between. And then the other type is bacteria that cannot use oxygen for metabolism, but they are not harmed by it. So basically they can survive in an environment that has oxygen, but they don't need it to survive. Those are called aerotolerant anaerobes. So once again, pretty easy to remember. They can tolerate an aerobic environment, but they are anaerobes. So pretty self-explanatory. The two main types of cell wall in bacteria. Those are the gram-positive and the gram-negative. And the way they figure out which one it is they do that staining with crystal violet and see what color it turns to figure out if it's the gram-positive and the gram-negative. But they have more differences besides that as well. The type of cell wall in bacteria is determined by, just like I said, the gram staining process. They do the staining, see what color it turns. Gram-positive cells consist of a thick layer of blank, a polymeric substance made of amino acids and sugars. This is one of the primary biological differences between them. That is peptidoglycan. Let's see, is there a picture of it? There is a picture of a prokaryotic cell in here. It's kind of what a prokaryotic cell typically looks like, so you can tell much simpler than the eukaryotic one. So the gram-positive one has a lot of peptidoglycan in his cell wall. In addition to peptidoglycan, the gram-positive cell wall also contains, so in gram-positive cells we also have this lipoteichoic acid. And the interesting thing about this, the lipoteichoic acid, so it's really only present in the gram-positive bacterial cells, but we're not quite sure exactly what it does or what purpose it serves just yet. We're still kind of figuring that out. The only thing we do know is that it can trigger an immune response in humans. So if you get infected with a gram-positive bacteria and you end up with some sort of an immune response to it, which I would hope you would, it's that lipoteichoic acid that's triggering that. But we're not really sure yet what purpose it actually serves for the bacteria itself. Now, gram-negative cell walls are very thin. They're much thinner than the gram-positive, and they also contain the peptidoglycan, but in much blank amounts than gram-positive. I pretty much said it already. They contain it in much smaller amounts than the gram-positive. Gram-negative bacteria also have outer membranes containing blank and blank. They have the phospholipids and the lipopolysaccharides. And those are the things, the lipopolysaccharides, that's the thing in the gram-negative bacteria that triggers the immune response in humans. Humans tend to have a very kind of inflammatory response to lipopolysaccharides. The ability of a cell to detect chemical stimuli and move toward or away from them is chemotaxis. And the way I remember this is kind of silly, but I think about the word taxi is in it, and so moving towards or away from something, moving away from the chemical. So once again, kind of a silly way of remembering it, but it works for me. Flagella are composed of blank, blank, and blank. That's a filament, a basal body, and a hook. Oh, this is already like 25 minutes long. I'll try to get through the rest of these more quickly. Sorry, I keep going on tangents. Many bacteria contain extrachromosomal material called plasmids. Plasmids also very interesting. I won't go on another tangent though. Blank carry DNA that is not necessary for the survival of the prokaryote, but may confer antibiotic resistance. That is also plasmids. That's actually what I was going to say is so interesting about them, is that so plasmids, they're not necessary for the bacteria to survive, but they carry certain traits, certain DNA that that tends to be very interesting, and one of them one of the things that ends up being in the plasmids a lot is the gene for antibiotic resistance. So that's something that can be carried in plasmids. Because binary fission requires blank events than mitosis, it can proceed more blank. So binary fission is one of the main ways that prokaryotic cells such as bacteria reproduce, and because it requires fewer events than mitosis, which is the way eukaryotic cells reproduce, it can proceed more rapidly, much more rapidly. There are actually certain bacteria that can reproduce once every 20 minutes, which is pretty astounding when you think about it. The stages of binary fission. So the three main stages are replication, invagination, and division. I do think there is a diagram of that in here. I remember seeing it. Oh, also really quick, this is the flagellum structure. I really like this picture. It looks like some sort of like Cthulhu monster came out of a lab and would be attacking someone. I don't know. It just kind of looks to me like some sort of a like really bad horror movie, you know, some monster in a lab. Where was I? Oh yeah, so this is the prokaryotic division right there. Plasmids may also contain certain blank, such as toxin production, or features that allow evasion of the host's immune system. They may also contain certain, we call those virulence factors. Ooh, spooky name. A subset of plasmids called blank are capable of integrating into the genome of the bacteria. Those are called episomes. Recombination processes that help increase bacterial diversity include blank, blank, and blank. So by what ways can they recombine? Those are transformation, conjugation, and transduction. Blank results from the integration of foreign genetic material into the host genome. That one is transformation. Blank is the bacterial form of mating, or sexual reproduction. That one is conjugation, which I think is probably the most interesting one. So more about conjugation. Two cells form a blank to facilitate the transfer of genetic material. They call that the conjugation bridge. There's also a picture of that in here. So basically, one of the cells, it has to be a male cell, has this little pilus, basically kind of like this little wiener sort of thing. I can't believe I just said that, but so they can extend that, and that's how they can reproduce by conjugation. And it has to be the male cell that does it. The transfer of genetic material is blank from blank to blank, kind of what I was just saying. So it's unidirectional from the donor male, which is considered positive, to the recipient female, which is considered negative. So we're going to only go that one way. It's unidirectional. The conjugation bridge is made from blank found on the donor male. That is the sex pili, that pilus, that kind of like, I guess, the distasteful way I described it is kind of that little wiener-like thing. To form the sex pili, bacteria must contain blank that contain the necessary gene. Let's contain the sex factors. Cells that have undergone bacterial conjugation are, this might be a term you've heard before, it's called high frequency of recombination, or that capital HFR, like that. A blank is a virus that carries genetic material from one bacterium to another. That is called a vector. Blank is the only genetic recombination process that requires a vector. What's the only one left of the three? That's transduction. Blank can accidentally incorporate a segment of host DNA and release this trapped DNA into a new host cell. Those little guys are bacteriophages, another kind of really fun looking thing. Blank are genetic elements capable of inserting and removing themselves from the genome. Those are transposons. In a new environment, bacteria first adapt to the new local conditions during this is going into kind of the life cycle of bacterium. So the first stage is the lag phase, when they're first adapting to a new environment. Rate of division increases, causing an exponential increase in the number of bacteria in this is the second phase, called the exponential or the log phase. Reduction of resources slows reproduction, resulting in that's when we get into the stationary phase. After bacteria have exceeded the ability of the environment, blank occurs. That's the final phase, the death phase. So once again, stages of bacterial growth. First we have the lag phase, then the log phase, then the stationary phase, and then the death phase. We'll go through those 4 stages. Viruses are composed of blank, blank, and sometimes blank. So they definitely have genetic material, they definitely have a protein coat, and then sometimes they have an envelope containing lipids, but they most definitely have the first 2, sometimes this. Because viruses cannot reproduce independently, they are considered, we call them obligate intracellular parasites. It's really a huge debate within the scientific community about whether viruses are in fact alive, because they have some characteristics of living organisms and then some characteristics of not living organisms. So that's something that I personally tend to kind of I have vacillated on a bit throughout my career in the sciences. There are certain times when, I hear an argument from someone, I'm just like, yeah, you know what? I think viruses are alive. And then I hear an argument from someone else and I'm just like, no, you know what? They're not alive. So it's something I personally go back and forth on a lot. Viruses must express and replicate genetic info within a blank because they lack ribosomes to carry out protein synthesis. Got to replicate within a host cell. That's part of what makes them a virus. A virus will replicate and produce viral progeny called blank, which can be released to infect additional cells. Those are the virions. All very sinister names for things with a virus, you notice. Blank are viruses that specifically target bacteria, and then those are the bacteriophages. Single-stranded RNA viruses may be blank or blank. They call them positive or negative. They fall into those 2 categories when they're single-stranded RNA viruses. Blank implies that the genome may be directly translated to functional proteins by the ribosomes of the host cell, just like mRNA. Those are the positive sense viruses. Blank RNA bacteria strands act as a template for synthesis of a complementary strand, which is used as a template for protein synthesis. And those ones are the other kind, the negative sense. Negative sense RNA viruses must carry an blank in the virion to ensure that the complementary strand is synthesized. They must carry an RNA replicase. Blank are enveloped, single-stranded RNA viruses. Those are we call retroviruses. You've probably heard that term before. Retroviruses contain blank, which synthesizes DNA from single-stranded RNA. They contain reverse transcriptase, and that's what allows them to do that. Stages of the viral life cycle. So we start with infection. Then we have the translation and progeny assembly. Then we have the progeny release. And then we go into either the lytic or the lysogenic cycle. And there is a really great diagram that I love in here of the lytic and lysogenic cycles. I think this diagram is so wonderful because I think it really is very detailed and then also shows the way the 2 interact very well. So I'm a big fan of this one. I'm thinking of like printing it off for my wall or something. I just think it's a really great, I put diagrams and crap like that on my wall because I'm a nerd. After translation, viral genetic material must blank to the correct location in the cell. So it's going to a different location, so it must translocate. Most DNA viruses must blank in order to be transcribed into mRNA. They need to enter the nucleus. That's where they got to go within the cell most of the time. Genetic material from blank stays in the cytoplasm where it is directly translated into protein by host cell ribosomes. Those are the protein-sense RNA viruses. Blank requires synthesis of a complementary RNA strand via RNA replicase, which can be translated to form protein. Those are the negative-sense RNA viruses. During a blank, the bacteriophage maximizes use of the cell with little regard for survival of the cell. So that's one of those two cycles. Which one is that? That one is the lytic cycle. Viruses in the lytic phase are called, we consider those viruses to be virulent. If a virus does not lyse its bacterium, it may integrate into the host cell as a blank or blank, initiating the blank. So that's when it can become what's called a provirus or prophage, and that's when it gets into the lysogenic cycle. And so it can go between these as well, as you can see from this diagram. It can go from the lytic cycle into the lysogenic. It can go from lytic, or it go into from lysogenic into lytic. It can go both ways. In the blank, the virus is replicated as the bacteria reproduces because it is now a part of the host's genome. So that's one of the advantages of the lysogenic cycle, is it doesn't destroy the host cell, and it can kind of take advantage of its metabolic processes. And blank are very small particles that can cause disease. We're talking about prions and viroids. Another couple things that I think are very, very interesting, particularly prions, but I won't go on to them too much. Blank are infectious proteins that cause disease by triggering misfolding of proteins, usually by conversion of an alpha helical to a beta sheet. Those are the prions. Probably the most famous example of prions that people were very scared of was the bovine spongiform, or the mad cow disease. I don't know if any of y'all really remember when there was the freak out about that. You probably don't if you're too much younger than I am, because I was probably only 12 or 13 years old when that was really in the news. But that was a huge topic in the news. The mad cow disease, and that is a prion. That's what it does. It triggers a misfolding of the protein, and that's what it does to people. Very, very scary disease. And then blank are very small pathogens consisting of very short, circular, single-stranded RNA that infect plants. And those ones are the viroids. So prions are the ones that infect animals, and then viroids are the ones that infect plants. So that's really, that was really the first chapter of this book, and I just made some flashcards from it, and we'll go ahead and cut this video short because I'm sure y'all are tired of listening to me talk, but hope this was helpful for someone, and good luck studying, everyone.